So yeah, I'm going to talk about the Biosamples database today. Just to introduce myself, my name is Tony Burdett, and I'm the technical coordinator in the Samples, Phenotypes and Ontologies team at the EBI. That means I'm the technical lead on the Biosamples database, but our team also has got several activities based around ontology tooling. So we develop OLS and Zooma and a few other tools and pipelines. I'm the sort of overseer, technical oversight on those tools as well, and I'm going to talk about a little bit about those in this talk. I'm also involved in the working group, the services working group for ELIXIR interoperability on Accelerate, and our team's tasked with delivering a GA4GH compliant metadata API. So I'm going to sort of briefly mention both of those as well in this talk. So yeah, just to kick off. This is a kind of slide of the data resources at EBI. I think people will have seen this slide probably before in these webinars. Biosamples is present in this space, in the systems space over on the left-hand side there. This slide shows you the range of services and data that you can access through the EBI. Biosamples is kind of key is sort of a key touching point to many of these resources. So it really serves as an integration point between a lot of archival and the assay databases. So samples present you a sort of integrated view over the European Nucleotide Archive, the Variation Archive. We've pulled data from Array Express, from PRIDE. There's connections between Biosamples and Biostudies, for example. So really, Biosamples connects to a lot of these resources, as we'll see a little bit as we go through this talk. So just to outline a little bit about what Biosamples actually is, it's a repository containing, at current count, more than 4.6 million samples, with over 15,000 metadata attributes. So just kind of as we've seen on the previous slide, most of the EBI data actually relates to one or more samples. So you'll see things like subjects, cells, tissues, ocean samples, microbiomes. Biosamples gives you this kind of connective bridge between the data that are generated over those samples in experiments and the sample metadata themselves. So really, it's kind of designed to give you an assay-independent space to work on sample information. So it's a component that enables cross-linking of assay metadata, of assay data to the metadata about the samples from which they're derived. So you can think of an example where we take reference sample data, so like Coriell cell lines or HapMap information, 1,000 genome samples. Many experiments have been done on that kind of data, and that assay data will be stored in EBI resources. Being able to link back to the original samples means that not only can you do queries across those samples, but also you can track the impact of high-profile samples or reference samples that are reused in lots of places. Some samples in Biosamples are only used once in a single experiment, but others, some of these high-profile ones, are used many thousands of times. So just to think a little bit about the problems that Biosamples are actually trying to solve, and just to summarize that a little bit. Effectively, we're taking data from biological samples across many different domains, across a wide variety of technologies that are currently archived in a variety of resources, and this data might be in kind of silos based on the type of data. So gene expression data might go to Array Express within the EBI, or GEO in NCBI, for example. Our mission is to try and connect that data back together and try and break those barriers down. So this enables a few things like finding data, querying across data, integrating data from different resources, sharing it between resources, analyzing and visualizing that data, all designed really to provide a better user experience when working with sample data. So if we look at the next slide, then this is kind of a sort of representation of the Biosamples ecosystem, really. So this is data where these so some of the places where Biosamples has data or connects to data. So some of it in this slide is pulled into Biosamples post submission. So for example, you might do a data submission to Array Express or PRIDE repositories. and Biosamples pulls that data downstream to try and connect it to other resources. In other cases, you might get data brokered to Biosamples from a direct submission. So if you want to submit sequencing or nucleotide information to ENA, you might submit that directly. At that point, the point of submission, the ENA would assign a Biosamples identifier and broker that information to us. And there's also a few large consortia that we work with, large groups or projects, and some examples of this are HIPSCI and EBIS, which are both induced pluripotent stem cell banking projects. And we work with those consortia to register samples and their metadata in advance. So that means we can do things like checking samples meet community-agreed metadata standards in advance before any experiments have been done on those samples, before any data has been generated. By pre-registering with Biosamples, you know that the metadata is in shape before you do any experiments. So we're still supporting that kind of pre-registration, or still improving the support for that kind of pre-registration with the archival resources. But you know, as we move forward, that we hope the uptake of those things will improve. So just to give you some numbers then, so this is kind of the species breakdown of the number of the different sorts of species that we have in Biosamples. So you can see, as you probably expect, human and mouse are the biggest, most studied species. So we've got more data for those species by far. The sort of that 14% other chunk indicates a huge variety of very small data sets for maybe one niche species. There's really a huge variety there that you can see. So I want to move on to cover some of the details about how the data is actually represented in Biosamples. So one thing that we're really interested in, as I've kind of alluded to so far, is that we're really interested in relationships between samples to help with linking data together and back to sort of the original sample, if you like. So we think about sample relationships. So sample A might be derived from sample B, or might be a child of that sample in some sort of pedigree relationship. Might be the same thing in some cases. And we think really hard about these kind of value-added pipelines about how to identify where samples have been reused or modified. So, for example, if in a lab as an experimenter you bought a cell line off the shelf from a stem cell banking consortia, you bought that, then kind of modified it in some way, used it in your experiment, and then submitted some data to an archival resource, we can track that all the way back to say, actually, this data was generated from this original cell line. And that gives us the opportunity to ask questions like, for example, I want to collect all of the gene expression data in any archival resource where that gene expression data it comes from a HeLa-derived cell line, for example. Or maybe we want all the gene expression data around all the sequences for HeLa-derived cell lines. So we can start to ask these sorts of questions and link data from assay databases together based on the Biosamples identifier. And we can also then track provenance as well. So we can say, okay, this sample has been derived from another sample by some experimental procedure if we need that level of expressivity. Linking is one key aspect, but probably more important is capturing good quality metadata. And metadata is critically important to this process. Our hope is that by collating sample information together in a single repository, we can try and encourage consistent annotation, consistent representation. We can try and promote standards that are available in communities. We can like show in context which standards are being more used in which fields of data, and then we can do things like aggregation and integration of otherwise completely disconnected data sets. And we can also do things like tracking high-profile reference samples, as I've mentioned previously. So this is how it kind of stacks out in practice. What we hope for, what we would like to see, is that the Biosamples data we pull really well-described, really semantically meaningful samples with like nicely typed attributes. So we say, okay, this sample came from a patient with a disease, diabetes, things like this. We can have these really nice, well-described samples. We hope that there's ontologies that cover the domain being studied. So if you're looking at some sort of plant sequencing experiment, for example, then maybe there's an ontology to describe the sequence and to describe the sort of species and the environment for the plant. And these are all kind of standard ontologies that are well used and agreed by communities up front. And then maybe sample metadata is annotated to those ontologies, and then consequently you can use that really rich, well annotated metadata in downstream analyses. So this is what we hope for. The reality often falls short of expectation. So what we end up with is a situation where you find there's free text everywhere, records explained in much detail in paragraphs of text. They might have been edited by several people in Excel, and there's a spreadsheet connected to a document somewhere else. Data's been pushed in and out of multiple different databases and got a little bit munged along the way. Maybe there's a local vocabulary either for that database or for a lab that's kind of doing the experiments or capturing the data in a spreadsheet. There's variation in spelling. Different people have different column headings with their own specific definitions. There's no versioning, and actually in the ontology space, there's many different ontologies for describing overlapping concepts. So there's many disease ontologies, for example. So we don't really very often get close to the ideal scenario. The bottom line is that data that's out there in the wild, in a real world, is actually just not that well harmonized. Biosamples trying really hard to deal with those problems in some sort of high throughput manner. There isn't really any substitute for actually getting your hands dirty and curating that data, but we can try to apply some big data techniques and ontologies to do the best cleanup that we can. So I just wanted to show a couple of sort of concrete examples of this then, basically. So if we drill into some of the challenges of the variability of the metadata, what we see here is kind of the word cloud for the attribute types that we have in biosamples. So these are the sort of column headings in a spreadsheet is what they would be. There's actually 15,000 different attribute types in biosamples, as I mentioned. And you can see some of the big ones that you'd expect. It's sort of the common things like disease state, like sex, organism part, organism. They're common, right? They're meaningful, and those are the ones that you'd expect. But there are definitely things that you can see in this slide that are overrepresented. So things like pathology report UUID, for example, illustrates this problem that I mentioned in the last slide about local vocabularies or attributes that are very specific to a single database. And if we move on to then take out one of those attribute types that we looked at, so this is the word cloud for the values associated with the attribute type disease state. Now there's actually, that's only one way of trying to express the disease on a sample. There's at least 150 other ways of creating a disease state, a disease attribute. So you'll see things like disease type, condition, diagnosis, many different ways of expressing the same information, but this is just looking at that one, and you see some big cancer type diseases, which isn't surprising based on some of the data that we've got. But what you see is that there are maybe 5,000 different values for disease state spread across over 1 million samples in biosamples. So at least 1 million samples have got a disease state attribute. There's 5,000 different diseases that they're looked at. So the challenge of trying to normalize this information is a really big one, but we know that there's lots of really good data. So we've got this kind of gold mine situation. We know there's lots of really nice kind of tight descriptions in there. They're very variable. Some are better quality than others. We just need to figure out a way to get in and extract this good stuff. So we have this kind of idea of having a sort of a mapping pipeline to take this raw metadata and turn it into some sort of explicit knowledge. It's a bit of a cyclic process rather than a linear one. I'll come on to that. But effectively, we put the data from biosamples into the top, push it down through this process. So it starts off by this what I'm calling here a data exploration process. This involves kind of looking at the data, trying to understand the scope, trying to understand the domain, check that we understand where the data is coming from, and trying to get a handle on whether it kind of connects well to data in that we already have in biosamples. Once we've done that, we push it through this kind of mining procedure. So we try and identify like kind of clusters of annotations or clusters of attributes. And we try and do a little bit of targeted cleanup to say, okay, well, when this set of data talks about gender, they mean the same thing as this other data set when they talk about sex. So, and we try and sort of identify these common cleanups, and we store those, and then we go through a process of ontology annotation where we map all these descriptions to ontology terms. So every sample then becomes tagged with a series of ontology terms. The cyclic nature of this comes in where this isn't linear. So in practice, we actually use information that's available in ontologies to explore the data. And trying to get a handle on understanding new data that we receive. And when new data comes in, we need to look at that and try and identify whether there are ontologies that describe that data already. And potentially both can feed back into the other. So, for example, we might need to say, actually, this is a new data type. There's no ontology for this. We need to start requesting new ontology terms to be able to describe that. So if we move on to the next slide. So yeah, just to summarize kind of what I've been talking about so far a little bit, we've got this really variable quality and annotation on data. And that's driven by the fact that biosamples accept sample data from a huge variety of sources. So most of that varies in quality. We need to try and look at the textual descriptions and try and figure out how best to normalize that data. And these things range from like very gold standard, highly curated data, right down to what I call system-generated noise. It's really just attributes that a database is stuck on through some submission system or through some automated pipeline process. We don't really know how useful any of that is. And the other thing to note is that there's a range of different ontologies that people use for annotation. So this shows a little bit of kind of disease space, so you can see just if you're trying to annotate your disease descriptions to ontologies, you could use MESH or Orphanet or EFO or HPO or ICD-10 or SNOMED or OMIM or the disease ontology. There's a huge variety of things. They all kind of overlap. They're good at some things and less good at others, but there's no way to kind of sort of produce a single kind of ontology that covers everyone's requirements. So you have to try and work out how to connect these things together. And in order to do this, it's really important to get it right early. So usually submitters of data, either sample data or assay data, have a good handle on the things that they're trying to say. So it's really important to capture good metadata about samples at the point of submission. So as I mentioned earlier, Biosamples is working with some larger consortia like EBISC and HIPSCI and FANG to try and do this. We also work with ENA to try and broker submissions, but we're trying to do this across all EBI submissions of future. So Biosamples is going to become more and more of a key component that happens really early in the point of submission to EBI. It's really important at that point to get good metadata and get it right. But we have challenges, right? So one of the challenges is to try and guide our users to give us good descriptions rather than force them. You can't always exactly know in advance what information about a sample users will want to give you. There's always something new comes along in biology. So really we want to say, okay, we think when you say this, we think you mean this. Is that correct? And sort of trying to guide users in the right way without necessarily forcing them into a vocabulary that they don't like or doesn't contain the terms that they need. And can we be responsive enough to new fields? So when a new type of experiment comes along or we have data that is for something that we've never seen before? How quickly can we respond to create new ontology terms or to adapt our templates, for example? And how do we validate effectively? Validation actually is something that's kind of harder than you might think. So covering a different range of vocabularies or different domains. So, for example, the term strain is a loaded one. So in a bacterial context, we might mean a bacterial strain, or we might mean a particular type of cell line, for example, that might be experimented on, and it means something completely different in the plant context when you're probably talking about a breed of a plant. But all of these fields will use the term strain in a submission. So how do we identify one way to validate the use of the term strain? And we're also, this is the point to mention, we're working quite closely with the Accelerate use case partners. So we've been having conversations with people in the areas of marine metagenomics, in plant space, in rare disease, and human access control data to ensure that we can try and build these kind of standards compliant validation or templates to try and say, actually, you've given us all of the data that we need to be able to say this is a good description of a sample. And there will be tough approaches that need to be taken. So can we say, okay, well, we're not going to accept this data because you haven't told us enough about it. But you know that's also a balancing act. Do we want to force people to not submit their data because we're too restrictive? I think that's something that we definitely don't want to do. So trying to find that balance between encouraging people to give you as much metadata they can without making it really prohibitively expensive for them to submit is a real challenge. And so this feeds me into talking a little bit more about ontologies, and I'm going to come on to that. We know that the data that we see, the metadata attributes that we see in biosamples, comes with this kind of long tail. So you see a few terms that are really, really heavily used. So some of the diseases might be well used, things like sex, male, or organism, Homo sapiens. You will see at the top end of this graph all of the time. Other things are much less frequently used or kind of unusually worded. So our challenge is to try and make sure we identify the terms that people use commonly, and we know how to annotate those without worrying about the like last 20 or 30% of the tail, which is things that have only ever been used once in one niche experiment. So this is kind of a process of like smelting down effectively. So we take that gold stuff that we found in our gold mine earlier on, we melt it down, we're trying to remove some of the kind of impurities and push more and more of the data that we see up towards the fat part of the graph and shorten that tail effectively. And so we have some tools to help us do this. So we make heavy use in biosamples of a tool that we've built in our team called Zooma, which is a tool designed to take textual descriptions, short textual descriptions, and map them to ontology terms. So just to blow that up a little bit, basically you can drop in some short descriptions, things like Crohn's disease, diabetes mellitus. And Zooma will then kind of use that to predict you a mapping to an ontology. There's some configuration that you can do. I don't want to talk too much about that today. I can take questions on that later if we need to. But the idea then is we say, actually, we're really confident in the mapping between the little description for Crohn's disease that you gave us and this ontology term, EFO384. We know that because this has been seen previously in other databases. Curators elsewhere have taken that textual description and mapped it to an ontology term. So there's real data backing this thing. So I think that's the key message to take away with Zooma, is it's not about looking at maps between data and labels in ontologies. It's about taking advantage of real data that's been curated, and ideally real data that's been curated and kind of looks a little bit like the data that you're trying to describe. And that's hugely beneficial over this kind of label matching process that you would see from an ontology, because just looking at ontologies, how do you know if people are using that ontology? or lots of other people using that ontology? Is it a community standard ontology, or is it just a student project? Was the ontology designed with my particular use case or my particular context in mind? Zooma is a tool where we're trying to capture that. So we're capturing the use and the provenance and the tracking of changes as well. So where ontologies evolve or data evolves, there's a challenge there to try and keep up with that. And Zooma gives you a little bit of a buffer to do that. So we make use of this tool called Zooma in our process. We also make heavy usage of the Ontology Lookup service, which I think has been a subject of a previous webinar. My colleague Simon Jupp in my team, I think, has already talked about it, so I don't want to go into too much detail. But the scenario here is effectively, if you can't find something in Zooma, if we, when we're describing biosamples data, can't find a term in Zooma that hasn't been curated elsewhere, we will go to the Ontology Lookup service because there are many more terms in OLS than those which have curated data for. But as we find terms in OLS and curate them and look at them within our team, or other curators look at them and add that data to Zooma, we kind of get better at predicting future mappings. And this is very much an effort that we're driving on at the minute. And then one other key component of this kind of ontology mapping pipeline that I want to talk about is Webulous, which is a tool that we've also developed in our team, designed to help us evolve ontologies. So, for example, when we find data that we've never seen, or find data, short metadata descriptions for terms that we've never seen before, we can't find them in Zooma, we can't find them in OLS. We have this kind of tool to say, actually, we don't really know what this thing that you're trying to describe is. Can you give us a little bit more information? And we'll turn it into an ontology term for you. So that's kind of where Webulous comes in, and this kind of helps us feed into that cyclic process where we're continually both mapping the data better, but also evolving the ontologies. And by taking these sample descriptions and mapping them well to ontologies, we get into sort of the value that we get from that. So the key one for biosamples is query expansion and some slightly more advanced stuff on top of that. But we can do these sorts of things. So we can say, actually, if a user comes along to biosamples, starts querying for all the inflammatory bowel disease samples, we can show them things. And I don't know if this came out a little bit small, but the top hit in this search result is actually for Crohn's disease. So a search for inflammatory bowel disease will return Crohn's because Crohn's is a specific type of inflammatory bowel disease. You'll also see hits in there for things like ulcerative colitis. So we might not be matching any of the words, but we can match the concepts because we know them from the ontology. And we can also do things like, I don't know if you can really see this, but we can show hyperlinks. So we can actually say things like, where you've got an attribute organism with a value Homo sapiens, we know the ontology term for that. So we can hyperlink out to that to say, Homo sapiens, if you need a bit more description, here it is. So this is really good for certain types of disease or phenotype. And we can link through to the Ontology Lookup service, and we can show the descriptions of those concepts there. But just on top of the things that we're doing in biosamples, they're kind of ontologies. This is a bit of an ontology cell. They add way more value than this. So smarter searching is the one that we're trying to provide functionality for in biosamples, but you can use ontologies as a key kind of component in data integration, data analysis, and visualization of your data. Which gets us to kind of like the end of the gold analogy. We've kind of taken our raw gold materials, we've melted it down, and we've turned it into something a bit more beautiful, something a bit more valuable, and hopefully something really highly useful. So that brings me on very quickly to how we plan to feed biosamples into GA4GH and what our future plans are. So this slide is really just kind of a list of things that we will do as part of our GA4GH program. We are charged with producing interoperable sample information under GA4GH Objective 4. So two key components of that are to implement an API into biosamples data using the GA4GH compliance schema, and the other component is to produce a sample identifier assignment system, which are both key components of biosamples right now. We also have an implementation of data ontology mapping tools to produce, and that very much ties in with this process that I've talked about, this kind of Zooma OLS-driven process. And then an implementation of GA4GH access tools and widgets for ontologies. And there's some little examples shown on that slide. This is a good time then to talk about the APIs into Biosamples. So we have quite rich data APIs into Biosamples. We can provide JSON and XML data, the sample description, and for the relationships between samples, as I've sort of mentioned. That's both relationships between samples and other samples, and relationships between samples and their data. So this sample has sequencing data in ENA. Example. And we can expand these APIs. That technically it should be relatively straightforward to expand these APIs to be compliant with GA4GH, and that gives us the ability to ask questions like, get me all the samples about inflammatory bowel disease, including Crohn's disease and ulcerative colitis, and then get me the links to the sequence data and gene expression data, and maybe some proteomics data for those samples as well. So as I mentioned, I think technically aligning Biosamples with GA4GH is actually relatively straightforward. The much bigger challenges are with the metadata and with the consistency of the metadata. As I've talked about, I think for GA4GH, this kind of prospective ontology mapping is really needed. And for GA4GH, there's this kind of natural focus on a subset of ontologies. And we should use them and focus on data that kind of looks like that. So sort of HPO, medical coding, kind of also anatomy and sequence ontologies. And we can take advantage of the work that we've done in Biosamples already by reusing things like Zooma. And I think there's a key lesson to be learned from Biosamples, which is about using existing ontology services and not heavily embedding semantics in yet another new solution. So by linking out to services like Zooma and like OLS, we can cross-link rather than sort of re-import. So a key problem that we've dealt with in the past is handling change. So where ontologies change, or labels in ontology terms change, or whether data changes, or ontologies are reorganized. We've actually, in our team, we've recently just completed a 3-year research project on this, and it's a really hard problem. It's best to avoid it altogether and rely on services that manage to wrangle that. So things like OLS will track change. It's better to link out to that service rather than re-embed. So that more or less brings me to the end of what I wanted to say there. I've got one quick slide. There's a sneak preview of a new Biosamples version. We have a beta release of a new Biosamples API. It's been released hopefully on October the 6th. Feel free to click around, try it out, send us any feedback. The links are there in the slides. And then just to sum up at the end then, so when we're taking data from a series of diverse data sets, actually getting the metadata to align is a real significant problem. In Biosamples, I think I've shown that we've got strategies for trying to align metadata and get the most out of the limited resources we have available. Absolutely key to get better submissions, ideally with prospective ontology mappings. We think it's better to guide people to give you good quality metadata rather than force them. And there's no substitutes for agreeing standards up front, agreeing terminologies up front, and domain expertise and curation. So I want one very quick on acknowledgements slide. So I need to thank all of the samples, phenotypes, and ontologies team led by Helen Parkinson, and especially Adam Falconbridge, Luca Karubin, Simon Jupp, Cyrus Santivijay, and Olga Vruzko have done most of the work that I've talked about here. There's some acknowledgement of our funders there as well, and I will take questions at the end.